Hello, phonetics, and welcome to 3.3 Meiosis with Sophie Stroiva, where we will be discussing the process of meiosis and some abnormalities that can occur from it. Of course, I have provided a notes guide, which you can find on Schoology. I know you love those. Fill those in as we go along. So before we begin, we need some understandings, right? And so in this topic, we have 8. One diploid nucleus divides by meiosis to produce 4 haploid nuclei. The halving of chromosome number allows a sexual life cycle with the fusion of gametes. DNA is replicated before meiosis so that the number of chromosomes consists of 2 sister chromatids. The early stages of meiosis involves pairing of homologous chromosomes and crossing over, followed by condensation. Orientation of pairs of homologous chromosomes prior to the separation is random. Separation of pairs of homologous chromosomes in the first division of meiosis halves the chromosomes number, and crossing over and random orientation promotes genetic variation, and fusion of gametes from different parents promote genetic variation as well. So now we move on to applications and skills. Non-disjunction can cause Down syndrome and some other chromosome abnormalities. Studies show that age of parents influences chances of non-disjunction. You have to describe methods used to obtain cells for karyotype analysis, i.e. chronic villus sampling or an amniocentesis, and you also need to know the associated risks. You have to be able to draw the stages of meiosis resulting from the formation of 4 haploid cells. And as you can see on your notes guide, you have separate boxes for each stage of meiosis. So let's break it down. The vast majority of cells in a person's body contains 46 chromosomes. For an individual to have only 46 chromosomes, each gamete, so what's that? Remember, an egg and a sperm must have 23 chromosomes each, so that when they fuse, they create a total of 46. To make gametes with only 23 chromosomes, a special process called reductive division must occur, which is theoretically just the splitting of DNA. Let's break it down a bit further, and we're going to look at meiosis and mitosis. So in mitosis, we have a diploid nuclei, and the formula used for this is 2N. And it results in 46 chromosomes in total, meaning 23 pairs. And in meiosis, it results in a haploid nuclei, and the formula followed for this is just N. So you have 23 chromosomes, which is half of one pair. It's important that you write this down, especially this part, into your notes and highlight key words. First, vocabulary review. The first word is chromosome, which is a thread-like structure, which pretty much just contains genetic information in the form of genes. Word 2, reductive division, very important, is when the chromosome number, which is initially 46, is split to 23, so from diploid to haploid. Diploid, again, means containing 2 complete sets of chromosomes, so 46 in total. Haploid is having a single set of unpaired chromosomes, which is 23 chromosomes. Then you have gamete, which is a mature form of a haploid cell, which can be given from the male as a sperm or the female as an egg. And then you have zygote, which is when those haploid cells fuse to create a diploid cell. Hey, what? DNA is replicated before meiosis? Yep, of course, this is why the chromosome resembles the letter X or H. So let's take a closer look. This also means that a single chromosome is made up of 2 sister chromatids joined in the middle by the centromere. So picture that in your head. In the early stages of cellular division, DNA undergoes condensation and the chromosomes coil up. So the pairing of homologous chromosomes and crossing over. So what is this end result that we're looking at? Diploid parent cell produces 4 haploid cells. Cool. So this requires the parent cell to divide twice. A key factor which distinguishes meiosis from mitosis is prophase 1, because at this point, there's an exchange of genetic material called crossing over, and this forms recombinant chromatids. So what's so special about crossing over, you may ask? Well, it allows maternal and paternal chromosomes to mix, resulting in genetic variation. This is one of the processes that create genetic variation, important to know, and meiosis 1 produces 2 cells. So next step, random orientation. So what is that? So during metaphase 1, when chromosomes line up by chance along the center of the equator, and in this case it's the vertical equator, this also increases genetic variation. It creates a low probability that a female and a male were to produce the exact same copies for a sperm and an egg. Only in the case of identical twins can a male and a female make the same offspring twice, and this only occurs because the two fetuses grow in one egg. So vocabulary review number 2. Guys, it's very important that you know this vocab so you can recognize these words on a test and it'd be easier to make the decision. So condensation, we all might know what that is, maybe from earth science, from chemistry, but it's when smaller things create bigger ones. So in the case of biology, you have, single strands of DNA forming chromosomes. Crossing over is just the exchange of genes between homologous pairs of chromosomes, and it results in a mixture from paternal and maternal characteristics for the offspring. Recombinant DNA chromatids, remember that's just a review of what we call the product after crossing over, and then genetic variation is just a permanent change in a chemical structure of chromosomes. So, the big question here is, how do we halve the number of chromosomes? Is it meiosis or mitosis? To determine this, we first must look at the process of, what is it? Meiosis. Of course, give yourself a pat on the back if you got that correct. So the first step is prophase 1. Chromosomes become visible as DNA becomes more compact. Homologous chromosomes pair up. One is from the mother and one is from the father. The magical process of what? Crossing over occurs, and spindle fibers are formed from microtubules. Now, if you look at this picture, it just shows you what crossing over really looks like. So take a second, pause this video, and draw this, this cell so that you know what prophase 1 would look like. So metaphase 1, it's when homologous chromosomes align across the cell's vertical equator. Remember, this occurs randomly, and the nuclear membrane disintegrates. This is a picture for you to draw. Anaphase 1, spindle fibers from the poles attach to the chromosomes and pull them in opposite poles of the cell. Remember, this just separates the DNA. Telophase 1, spindles and the spindle fibers disintegrate, chromosomes uncoil, and the nuclear membrane forms most of the time. It's important that you remember that most plants do not have this stage because at the end, they have cell walls. In animals, they have cytokinesis, which is when the cytoplasm splits. And now, at the end of meiosis 1, you have 2 haploid cells. Vocabulary review number 3. I promise, guys, this is the last vocab review. Again, very important to be familiar with these words. So cytokinesis, as we just talked about, it's a splitting of the cytoplasm, which pretty much just divides the cell into 2. Homologous chromosomes, maternal and paternal chromosomes that have genes at the same loci means location, but possibly different alleles of those genes, so dominant versus recessive. So guys, are you ready for meiosis part 2? Yay! So, yes, prophase 2, the process pretty much starts over. The DNA condenses into visible chromosomes once again, and new meiotic spindle fibers are produced. Go ahead, draw this. You can look at what the difference would be from the previous. Metaphase 2, the nuclear membrane disintegrates. This time, when the chromosomes align, they align along the horizontal equator. Highlight this, horizontal. Spindle fibers form from opposite poles attached to the sister chromatids at the centromeres. Anaphase 2. So the centromeres of each chromosome split, releasing each sister chromatid as an individual chromosome. And yes, an individual segment of DNA is still labeled as a chromosome, no matter its amount. So whether it's a pair or half of a pair, which is a sister chromatid, or half of a sister chromatid. Of course, the spindle fibers pull the individual chromatids towards opposite ends of the cell. Because of random orientation, the chromatids form new daughter cells. So let's discuss plants versus animals. In plants, the cell membrane pitches off in the middle because we don't have a cell wall, whereas plants that do, they form new cell plates to split the cells into 4. Now we go to telophase 2. Yes, at this point, the chromosomes unwind once again, and they form strands of DNA, and the nuclear envelope forms again around the haploid cells, preparing them for cytokinesis. We should all know what cytokinesis means at this point. Remember, splitting of the cytoplasm. You know how we have the number of chromosomes? Remember, we have, what is this fantastic process called? Reductive division. Yay, if you got that, you are a genius. So, going off to the second part of the presentation where we talk about some abnormalities that can occur with meiosis, we have fertilization and, variation. So, what causes genetic variation? Remember, random orientation and fertilization. Fertilization we haven't talked about yet, but it's when there's a unique combination between the maternal and paternal DNA. So, how do we calculate, how many different possible combinations there are? So, because each gamete, which remember is an egg and a cell, have 23 chromosomes, we follow the formula 2n. So we raise the power of 2 to 23, n equals the number of chromosomes. So this leads to 8,388,608 possible variations. But remember, this only accounts for one gamete. Now multiply this number by 2. You get 16,777,216 different variations. So, is meiosis a perfect cycle? What do you guys think? I think definitely not. Sometimes a child can receive an atypical number of chromosomes, 47 instead of 46, for example. And this actually causes Down syndrome. It's when a woman gives 2 chromosomes and a male gives 1, total of 3 chromosomes on the 21st loci. So this is also referred to as trisomy 321. This results because of non-disjunction when the chromosomes fail to separate during anaphase 1. So speaking of Down syndrome, epidemiological studies have shown that age of a mother can have heavy influence on whether or not a child will have Down syndrome. So what kind of advice can you give to an expecting mother over the age of 25? Well, for one, have an amniocentesis or a chronic villus sample. What are those, you're probably asking? Well, amniocentesis is the use of a hypodermic needle to extract amniotic fluid around the developing baby. This liquid is then used for a karyotype. Yes, this is a very expensive and, luxurious process, but it has been proven successful in catching Down syndrome, along with, of course, other sicknesses early on. It has a 1% rate of miscarriage and can be done during the 15th week of pregnancy. Compared to chronic villus sampling, which is obtaining a tissue sample from the placenta's finger-like projections, it's a bit more invasive and therefore has a 2% rate of miscarriage. But the plus side of this, it can be done at the 10th week of pregnancy. So these images, on your notes sheet, it's asking you to label which one's an amniocentesis and which one's a chronic villus sample. So based from these pictures, you can tell that the first one with a hypodermic needle is amniocentesis. The second slide is the chronic villus sample, and you can see it is a bit more invasive. Okay, so try this yourself. So at this point, you can slow down the video and actually do these. These will show you what you really know. So can you draw these stages of meiosis from memory? Do you know the 2 things that lead to genetic variation and where they occur? Can you explain what non-disjunction is? Guys, thank you for watching. I hope you enjoyed this video. All of the questions at the end, the answers are said throughout the video. Thank you and have fun studying and getting a 100 on that test.